Okay, so let's move on then without any questions, and we'll talk about figures. So you guys know that your next assignment is to take that first draft of your background and significance section and add figures. So we are going to talk for the rest of the class today on figures. And then what I'd love for you guys to do is, based on what we're doing here, to, in your breakout sections tonight, maybe talk about where a figure might be effective. Okay? Okay. So let's first talk about why figures are important. So you've heard the term, a picture is worth 1,000 words, and in chemistry and science, that is absolutely true. So let's do this example together in class. I'm going to read part of this. I don't want to read this whole thing because it'll take forever, but let's read this as an example of a picture is worth 1,000 words. Here's the paragraph. Enter the maze and turn right at the first intersection, being sure to avoid paths leading left or straight. Follow the pathway until the path turns right. Follow a turn to prevent hitting the wall. Shortly, turn left, following another immediate left. This is just tedious. And nobody wants to read this because we'd rather look at this picture where you can see we enter the maze, we turn left at the first intersection, being sure to avoid paths going straight or left. We follow the path until it turns right. Then we follow until we hit the wall, and then we turn left. So you can see that in this case, this maze picture is much more effective than the description in the text that we would have to write. There are many times when a figure like this is going to be much more effective in displaying and conveying information than text. So let's talk about a figure and what it can do for you. So there are two principles I want you to take home with you about figures. The first is that the figure should explain, it should convey an idea that's just basically too long to put into the text. And that's a beautiful example of this maze example. Having to write this tedious text it's just not as effective as showing the picture. And then please remember that each figure that you create, or table, we'll talk about tables also, but each table or text that you create needs a purpose. What is it telling the reader that is too complicated to put into the text? So kind of to give you an idea of what kind of information would be appropriate for a text, for a table, or for a figure, I took some inspiration from this webpage called editage.com. It's got a lot of information, and this was from one of the blogs in that. So I give you the link right here. But in that blog, they talk a little bit about what would be appropriate for these three different ways of depicting information. Text is really appropriate when you have simple data or simple relationships. You have to talk about a number, or you're talking about the relationship between this metal and this metal, just two, or maybe three, but not very simple relationships. Tables, on the other hand, you would want to use when you're comparing and contrasting several different characteristics or more complex data. And you want to see relationships between, kind of seeing the actual characteristics on one table. Now, on the other hand, figures are good for talking about trends and relationships among data or among characteristics. So you can have a visual explanation that can show a sequence of events, procedures, structures, or trends, okay? So these are kind of what you would want to be looking for as you start writing your papers. Now, where is it typical, particularly in a proposal or a research paper, to use these kinds of formats? So that's what I'll have. I have a little list here, and this is not a completely thorough list. I just thought of some of the things I felt like you would see in your proposal and in a research paper. I will talk a little bit about research papers here too. So when you do get to the point of writing your first research paper, you can come back to our class and also help you write those figures. So a table is really good when you have a list of reagents, a list of properties, and maybe you have a few different properties you want to compare and contrast. And I'll give you an example of how to make one of these, and we're just going to do this in the Microsoft Word package because I think that's the easiest place to do it. Figures then are going to be really appropriate for these other kinds of relationships. Complicated relationships like in pathways or between molecules. It's really hard to explain a chemical structure or a biological protein structure. So we're going to use figures for that. Anytime you have a workflow of experiments where you want to show kind of consecutive experiments and how they would relate to one another, how they would compare. That's where you would want to use a figure. And then something that's maybe not as appropriate for your independent proposal, but would be for your prospectus and for your future research papers, would be to show the relationships between multi-component data. And so I'm going to divide this rest of this module into talking about each of these 5 different types of figures, okay? So let's first talk about a list of reagents. So a list of reagents will be really good if you want to show kind of comparing, contrasting, and absolute data for something. And so I thought, going back to the outline of our research proposal for our COVID-19 project, I thought I would highlight and make a table that really shows the advantages and disadvantages of PCR versus antibody testing, because that's really what we're comparing our COVID-19 project. So for that, we're really going to use Microsoft Word, and I thought I would just show you really quick how I would do that. Maybe you guys know Microsoft Word really well, and if you do, just give me a little chat, put in the chat that I can move on. But basically, when I put a table into Microsoft Word, I would always insert it as a text box, just because you can wrap your text around it. I would always start my text box, and I prefer not to have an outline of my figures and tables, but that's up to you. You can have this. So you would write, table 1, and we're going to talk about, the detection methods for viruses. Well, of course, I'll say viral infection. And then here is where we will insert our table. So we're going to insert our table here, and I'm not sure how many columns. I know I need more than 2 rows. So we'll put in my column, and then we can talk about the method here. So we'll do PCR as one of those methods. We'll do the antibody testing as one of those methods. And then you can put in, we can have the advantages here, and we can put disadvantages. But you see what I mean? So you can fill this in. And then usually you want to have a footnote, so you can explain maybe where you got maybe references would be here. Okay, anyway, you get the idea. If you want a footnote, you can put a footnote here at the bottom. So I would probably use like a little asterisk here, and you can write a footnote in case you have something you need to tell them, tell the audience that's specific to this particular table. And I'm going to highlight this all, and I guess it made it in Cabri, which I don't like, so I'm going to change it to Arial. And please remember, any of these figures we're making here, you do not have to have 11 or 12 point. You are welcome in a figure to use 8-point font. Nothing smaller than 8-point font. 8-point font is the smallest you can use in a figure or table, but 8-point font is fine here. Okay, so with that little tutorial, let's go back to our module. So hopefully that helped you show how to do a table. Oh, let me show you that in this table, one of the nice things about putting it in the text box is now you can wrap it. So if I want to wrap this text, I can make it square, and now my text will go around my table. So that way I can use the space of my proposal the best I can. Okay. Now let's move on. So that's a list of reagent properties, characteristics. The next one I'll talk about are complicated relationships. And for this one, I'm not going to give you a tutorial, but I will highlight one proposal. So they have really nice sample proposals on the National Institute of Allergy and Infectious Disease website. Here's the link. If you haven't already, I strongly, strongly encourage you to take a look at some of these proposals so that you can learn the way that really good proposals are written. It's always good to look at good examples of good writing, right? So this is one. This is actually from Professor Tom Muir, who's a chemist at Princeton. And one of the first figure in his proposal, this example proposal on this NIAID website, is this very complicated looking graphic. And this is showing kind of the background information of how virulence works in the context of Staph aureus. This proposal is about understanding how Staph aureus works. And so talking about now the mechanism of that organism is what they're planning to do here. And you can see that this particular figure shows how this protein AGR is exported as a peptide, which can then signal to its own cell membrane to activate a response in the cell. And so this figure is really very nice here because it shows relationships between the protein, AGRD, the cognate peptide that's produced, and how that results in a change in cell signaling. And then, of course, you can show all the sequences of these peptides, and these are the ones they're going to be making in the proposal. So this is a particularly nice example because it shows how one picture can give you an idea of how all the relationships between, in this case, peptides and proteins how it works. Okay. So if you have complicated relationships between players, reagents, metals, proteins, whatever your proposal is about, this would be a great way to depict that. Okay, so in this case, you would make one of these figures in PowerPoint or Illustrator and Photoshop, and we will give you tutorials on all of those. The second figure would be a chemical or a biological structure. So I showed two figures from that same Tom Muir proposal, where we can see that, you know, it's hard to talk about in text a chemical structure. Just use ChemDraw, and Ed will give you a nice tutorial on ChemDraw in a bit. If you have a crystal structure of a protein or an image of a nanoparticle or something like that, you really can't do that in text. So that's where you would create a pretty figure. And again, we'll do that again in ChemDraw for structures. There's a program that we aren't going to be able to have time to go through called JMOL, which is available through the PDB website, where you get all crystal structures. You can use that to make these beautiful crystal structures of proteins, for those of you that will do that. Okay, so that's structures. You need pictures for structures. I'll do a little tutorial here on how to use PowerPoint by talking about how to develop, how to use a workflow. So I thought what I would do here, so a workflow is going to be sort of the logic behind how you would do a experimental reagent. And I thought what I would do, and hopefully this will be helpful, I thought I would basically show you kind of how to remake this scheme 1 in our COVID-19 inspiration paper using PowerPoint. So in PowerPoint, again, if you guys feel like you know how to do this already, just send me a quick note and I will move on so that we can get to other things. But I thought this would be a helpful way to just show you kind of how this works. So if I'm going to reproduce, so I'm going to kind of use as inspiration this scheme 1. If I was to reproduce this, I'm going to insert a variety of shapes. So let's say that I'm going to use a bead. I'll use that as a magnetic bead here. And I'm going to add also, I'm going to label this as a magnetic bead using a text box because I want to make sure that my, my audience knows what I'm talking about. So this is a magnetic bead. Okay, and remember, I'm going to be using 8-point Arial font here, so I'm going to change that right now. Okay, so there's my magnetic bead. I'm also going to need an antibody because remember, the thing that we're doing here is we're making these antibody-coated beads. So I'm going to take my my bead and I'm going to add. So I'm going to put a little plus sign there. I'm going to add a shape that looks like an antibody, and an antibody, it looks kind of like a Y, and actually I think the best kind of Y would be in ChemDraw, so I'll show you that in a minute. But I'm going to use that, then I'm going to insert now, the reaction will be an arrow, so I'm going to draw an arrow here so that I can create now my bead. Oh, and something I should mention, is that you want to make sure that you draw your figure to scale. Oops. You want to make sure you draw your figure to scale. So for example, if we go back and look at my Word document that I'm going to be inserting this into, notice that the size of my page is about 7 inches wide, right? So if I want my figure to be about half of that page, I want to make sure that I make it only about 3.5 inches wide. So when I make this figure, I maybe want to blow it up so that I can make sure that the figure is the right size for my document. So this is like 1, 2, 3. I really only want my figure to be up to about this 1/3 inch. So please, you know, keep that in mind as you're making your figure. You want to make it to scale so that when you add it back into Word, it's not too big or too little. So maybe I need to put my magnetic bead above, because it seems like I'm going to have too wide a figure. And like that arrow is way too wide, so I need to make that a little bit smaller so that I can scrunch everything into my half a, you know, kind of a one column. So here's my bead, and then I'm going to take this and add my virus. I'm going to do this quickly because I'm running out of time, but now we can take our viral particle now that we've made our beads with our antibodies. Here's our virus. And now we're going to take our bead and let's see, and we're going to have, remember the whole point of our method is that it aggregates these beads. So I'm going to take this bead and another bead, and I'm going to aggregate them along with my virus. And if I had more time, I would make little pretty pictures. If you guys want me to, you guys, I can finish this figure up later and show it to you. Okay. Okay, but anyway, then what you can do is there's actually two choices here in terms of how to get it into your Word document. I would highly recommend you group all of this and then move it into your Word document. And then when you put it into your Word document, you're going to want to add a figure legend. So again, you're going to insert a text box. So that'll be your figure legend. And then before you, and again, that can be Arial 8-point font. Before then you move it around in your document, because you want it to wrap test, you're going to take those two, you're going to then group them. Oops, sorry. You're going to group them. And then once you group them, like we did before, we can wrap the text square, and now you can add that to your document, and you can see that the text wraps around it very nicely. So now I can make my table a little bit smaller, and now you can see that my figure and my table wrap around nicely. Okay. All right, I've taken up too much time with that. So let me finish up this lecture by talking about multi-component data. And I'm really running out of time here. So I really want to show you this video, but I guess I'm not going to. This is a really nice video that outside of class, why don't you, if you have a bit of time, I really highly recommend that you talk about it. They talk about, this video is in terms of mouse studies, but I think it's really, really helpful to show how to organize data. So this is multi-component data that's maybe not appropriate for your independent proposal. You won't have any data. But when it comes to your prospectus or your research papers, this kind of organization of data is helpful. So, and this is a really nice video. So I don't have time to talk about it. I won't show it now. It's about a 3-minute video. But let me tell you that there are a lot of guidelines for how you can write and make a nice figure. So I took this guidelines from JVC. They have some beautiful guidelines for how to make a figure. Tell me what you think is wrong with this one. Any idea? Well, there's some things I can't read. And that would be the axes, right? Do you see how tiny, tiny, tiny the axis is? The font is? Please don't make that mistake. A good example here is where all of the sizes of the fonts are pretty similar. And remember, no less than 8-point font, right? Because we want a big thing. We want to be able to see it. Here's another example of maybe an incorrect use of data. Notice here, this is a patient-reported pain score for rheumatoid arthritis. This is their pain threshold over age group. And notice that they have this data plotted like there's a relationship between each age group, which is not true. There's no relationship between, like, why they drew a line between all of this data makes no sense, right? There's no reason that there would be a line or a curve or whatever, because these are 4 separate groups that are reporting on their pain. A much more appropriate way of depicting this data would be histograms and columns, because it doesn't imply that there's a relationship between the data. So the point here is that you really want to depict the data accurately without trying to overinterpret. And in fact, I really enjoyed reading this paper that I got this example from. This is a really, really nice paper that it's got a fun name. Graphs, tables, and figures in scientific publication, the good, the bad, and the not to, and how not to be ugly. and how not to be the latter. It's really a nicely done paper and shows kind of good ways to present data. And again, when you guys get to the point where you're writing your first paper, please, please, please take a look at this really nice article. Okay, so with that, let me just end by saying that your next homework assignment is to take that same background and significance section that you turned in today and adding figures so that you can make those points that you're trying to highlight in your background and significant even better. This is of course posted in Canvas, so I won't go through it, and here's your references. So I will stop there. Any last questions before I turn the host over to Ed?